It's Friday, 8. Again, good morning, everyone. I am in non-disclosed location under house arrest, but delighted to be with you all. And we have, as always, Friday, and 8:30 we have celebration of vision research. And today is not exception. I'm delighted to have Bryan joining list of speakers. And also as a reminder, you guys going to get a very soon invitation for our party at Arvo on Sunday. So keep your calendar clean. It's going to be better than Hopkins, Harvard, UCLA, Stanford, whatever it is. It's going to be a fantastic reception to all of you, all friends of this program as well. With that, let me just start with introduction of Bryan.
All right, you ready, Chris?
Yeah.
Okay. I see.
Good morning and welcome to the Distinguished Speaker Seminar Series of the Gavin Herbert Institute at UC Irvine. My name is Deepak Puria and I'm a project scientist in the lab of Vladimir Kafalov at UC Irvine. Today, I have the pleasure to introduce our seminar speaker, Dr. Bryan W. Jones, who will be delivering a talk about retinal connectomics and retinal pathoconnectomics. Dr. Jones is an associate professor at the Department of Ophthalmology and Visual Sciences, University of Utah Moran Eye Center at the Salt Lake City. He acquired a BS in Biology and Chemistry in 1996 and a PhD in Neurophysiology in 2003 from the University of Utah. During his graduate studies, he studied the molecular phenotypes of ganglion cells and the effects of photoreceptor degeneration on retinal structure. He then pursued his postdoctoral training in cell biology at the Huntsman Cancer Institute in 2003 and in ophthalmology at Moran Eye Center in 2004 at the University of Utah. Since 2005, he has been a faculty member at Moran Eye Center at the University of Utah. Dr. Jones is a recipient of several prestigious awards and honors. A Research to Prevent Blindness Career Development Award in 2006, the State of Utah Innovation Award in 2010, a Welcome Image Award from the Wellcome Trust UK in 2015, are just to name a few. He also won the NSF and FESIP Scientific Visualization Challenge in 2012 and 13. Dr. Jones has been extremely productive and has more than 75 peer-reviewed publications. His research encompasses neural plasticity, connectomics, retinal circuitry modeling, and pathophysiology of retinal degenerative diseases. Along with Professor Robert E. Mark, the Director of Research at Moran Eye Center, he has been extensively involved in the development of retinal connectome in healthy and diseased retina. Apart from being a researcher, Dr. Jones is also an avid photographer with a keen interest in invertebrate photography. His photography work has been published and telecasted in international media like National Geographic, Science Friday, The Washington Post, the Smithsonian, and The Guardian of UK. So without further delay, let's give a very warm welcome to Dr. Bryan W. Jones.
Thank you so much. Let me share my screen here. All right. Can you see that?
Yes.
Okay.
Yes.
Thank you for the introduction. I really appreciate it. To start out with, I'd like to thank everyone here. I'd like to thank Chris and Groznia for inviting me to participate in the UCI ophthalmology research seminars. I can't possibly relate how invaluable this seminar series was for the entire vision research community, all through the dark days of the COVID pandemic, and now it's sort of become a bit of an institution. So, thank you, Chris and Groznia, for virtually hosting me and allowing me to be a part of the series and share some of the work out of my lab. I have no financial disclosures. So just about everybody here, either physically or virtually, has some familiarization with the retina, so I won't belabor an introduction to this tissue lining the back of the eye, other than to say that this is a combined layer cake-like structure with sensors at the back and compute layers underneath. We're going to rotate the retina so that all the histologic images showing photoreceptors in this talk will at least be sort of photoreceptors up in the fashion of basic science rather than clinical imaging perspectives, unless we're looking en face horizontal sections through the retina. So we, as a community, have been studying retinas for 130 years or more. This classic and beautiful work by Ramon y Cajal in 1892 shows the neurons of the retina and the approximate laminations. Interestingly, Nico Cuenca at the University of Alicante is working with the Cajal Museum and was showing me plates where Cajal drew little arrows next to the neurons, illustrating the direction of information flow. And Jean-Marc showed last week in the seminar series, Cajal also illustrated the concept of convergence and seemed to understand implicitly that retinal neurons were directional and computational all the way back then, which is kind of humbling as we're still trying to figure this stuff out today. And while Cajal understood that these neurons were wired together, what he nor more modern neuroscientists understood for a while was how complex the retinal interactions are, right? So these connections in retina have been the focus of intense study from Cajal's day to so many of our friends and colleagues over time, and many others with more recent efforts through modern anatomical techniques such as confocal and then super resolution microscopy and now connectomics approaches in an attempt to figure out the flow of information in retina. It turns out retina is a wonderfully complex tissue with many cellular superclasses and many more subclasses of glial, horizontal, vascular, amacrine, bipolar, and ganglion cells. Each one of these cell classes has a unique relationship with the retinal network associated with it. And the thing that's always sort of like boggled my mind is that here in 2023, you know, after all these years of neuroanatomical and electrophysiological research, we neither know precisely how information is processed in complex neural networks, nor do we understand even the specific topology that defines these networks. And the interesting thing for me is that the path that current takes from photoreceptors at the photoreceptor terminal through the circuit topology of all of these complex networks gives rise to all the primitives that enable the calculations for visual and non-visual components of, you know, what we know as vision. Retinal connectomics, or the precise study of how these networks interact and are connected, offers us a roadmap to these network topologies and understanding of how retinal degenerative diseases alter those network topologies. And we believe that understanding the complexity of neural pathways in normal and disease states and how they function and fail is really critical to innovation in science, in blinding disease, and at the least it's a convenient framework to start hanging on all of the other electrophysiology, genetics, expression data, and even psychophysics data upon. Hopefully I can convince you of some of that today. So these algorithms or computations are really what defines the emergent phenomena that we can observe. Encoding of luminance, color, velocity, vector orientation, selectivity, circadian encoding, adaptation, sensitization, texture, and more. And there's lots of ways that we can query these complex assemblies of circuits to try and pull them apart, including this certainly non-exhaustive list here of anatomy, physiology, biochemistry, pharmacology, immunology, molecular biology, genetics, psychophysics, et cetera. And there's different experimental approaches to all of these disciplines. Today, I'm going to present to you several of these and explore how we're trying to figure out how the retina is wired and how it comes apart in disease, with connectomics serving as the framework that we hang it all upon. This is where I plug Greg Schwartz's excellent book on retinal computation. If you've not read it, it's a fun and highly recommended read. So if I'm interested in neural circuits, why retina? Lots of reasons. But first, I mean, so much of neuroscience has been pioneered in retina. Retina is a sophisticated signal processing device. It's accessible and has historically presented as an excellent framework with which to understand how neural principles are established and function. You know, and the other advantage is that they're like these remarkably compact network topologies, all contained within a few hundred microns, as I'll show here in a minute. To do the same kind of work in brain, you've got to go much bigger distances, and the technological limitations behind that can be substantial. So if this is a pyramidal cell on the right, as typified by what you'd find in cortex, and this is a bipolar cell, like what you'd find in retina, they both cover an area that's involved in circuitry that performs computations. At the scale of the pyramidal cell here, the trick is, while pyramidal cells can be rather large, bipolar cell geometries subtend a much smaller distance. And according to the Beaver Dam study and a conversation I had with Joe Carroll the other day, the mean thickness of human retina is around 334 microns, giving us a compact system where all the circuitry is available that performs all the calculations or algorithmic computation behind the calculated features that we just talked about, right? So if you want to understand neural systems and you want to understand how they compute information, retina is a pretty good system to do it in. So even though retinal connectomics as a discipline effectively pioneered much of the approaches that we're going to talk about here today, it's rapidly becoming a smaller subcategory of a much larger emerging field of volume electron microscopy, or volume EM, as it's becoming known. It's simply the application of large-scale ultrastructural approaches to reconstructing volumes of tissue or things, because people are interested in these approaches in material science now too. Modern computational approaches basically allow this to be done with far greater scales than were possible even a decade ago, as infrastructure costs and computational storage have plummeted. But I mean, the interesting thing is, for as long as people have been able to image ultrastructure, they've been looking at how they can go bigger to tell more of the story than they're able to see when you're looking through into this little, glowing chamber in an electron microscope. So people have used projections and subsampling for many decades in an attempt to understand neural circuits in retina, but the ability to systematically go through and serial section and reconstruct large volumes of tissue is a more recent phenomenon, right? As you can see from this paper, it's not to say that earlier efforts were not heroic. This paper, it might be the first volume EM-related paper in the literature. But this was 1958 when they looked at guinea pig eye and did a serial section reconstruction where they took 40 serial sections and reconstructed them. From there, small-scale serial to EM volumetric reconstruction of retinal tissues taught us about synaptic relationships of narrow field amacrine cells as well as bipolar and midget ganglion cells in the primate retina. Certainly the circuitry of cat A2 amacrine cell was first appreciated by these approaches and work by Kolb and Famiglietti in the 1970s. And then Kolb and West partially reconstructed interplexiform cells in human retina and demonstrated that bipolar cell axons make most of the ribbon output synapses to ganglion cell dendrites through her work in serial to EM and 3D reconstruction of cone bipolar cell processes. And then by 91, Helga and de Korver, I forget what her first name was, demonstrated that exclusive one-to-one relationships in midget pathways of primate retina, again through serial section to EM in primate paraphobia. Enrique Stratoy, working in Ray Deschaux' lab back in 1992, demonstrated beautiful partial ultrastructural reconstructions of A2 amacrine cells with gap junctions. This A2 stuff will be important in a couple of minutes here. And there's a ton of other folks that have also created volumes to explore ultrastructural circuitry over time. including Dennis Dacey, David Calkins, Peter Sterling, Heinz Vesely, Silke Haverkamp, Carl Klug, Yoshihiko Tsukamoto, Naoko Omi, David Burson, Kathy Morgans, the Knightses up at University of Washington, and many more. And just last night, I ran into this gorgeous paper from Gil Torton and my friends Steve Fisher, Kenyon Lindbergh, Gabe Luna, Mark Ellisman, Guy Perkins, and David Williams, exploring the ultrastructure of the normal rod photoreceptor synapse and how that changes due to retinal detachment. So I don't know how well this video is going to come across Zoom, but I'll try and play it anyhow. I hope you can see this. You have to appreciate how hard this sort of thing was in the 1950s up through the 1990s. In order to do this kind of reconstruction at the resolution desired to be able to resolve synapses and gap junctions and cover large areas of tissue, you had to mosaic images. And back then, electron microscopy data was captured on film and then enlarged onto photographic paper. And because of the distortions associated with TEM data, effectively there's a magnification astigmatism embedded within every image. So to make the mosaics work, you spend a lot of time with scissors and X-Acto knives and Scotch tape to get all the membranes to line up. And then if you really wanted to be clever, your correlative, your clem imaging or correlative light microscopy and ultrastructure or immunogold could be overlaying with cellophane sheets like I'm showing here that you can draw upon. So the folks that think that clem is a brand new technology are a little mistaken. So the idea of going bigger and creating a complete synaptic resolution map of retina is not a new idea. This goes all the way back to Clyde Oyster, famous for this text, who was talking to my PhD mentor, Robert Mark, back in 1978 about a complete retinal network map. They knew that the whole photographic paper mosaic thing just wouldn't scale and they'd have to use computers. But there were lots of problems in the late 1970s that prevented this from happening. Notably, such a project would have required lots of computational space. And in 1979, this was the state of the art in storage, the IBM 62 PC hard drive. It stored 65 megabytes of data and sold for $20,000. That's $89,534 in inflation-adjusted dollars for 2023. And the nominal size of our first retinal connectome was around 16 terabytes, which works out to about $353 billion in 1979, or $1.483 trillion in 2023. So fast forward a few years, and Robert and I had been talking over coffee about the idea to make this work. Robert said, "Do you thought we should do it?" I replied something to the effect of, "We can't afford the hard drives." To which Robert responded, "Right now we can't," but he'd been plotting out the price of hard drives over time. And he said, "If we start now, in a couple of years, we'll be able to afford it on extramural NIH funding." So it wasn't roughly until 2007 or 2008 when hard drive prices dropped low enough for NIH-funded labs to just be able to afford to store the data required of connectomics volumes. So you can ask yourself, what drives these storage and computational needs, right? And part of the crucial calculation that you need to make when deciding on making a connectome or a volume EM database is one of resolution. How much resolution do you need to be able to see the smallest thing necessary for your data? And in retina, for us, that was synapses and gap junctions. And Kristen Harris was working in hippocampus, and she was one of the first people to think about how to digitally represent synaptic topologies. In 1994, she demonstrated that you basically needed nominally 2 nanometers per pixel to resolve all the synapses and make a good guess at gap junctions. So we called that the gospel of Kristen, and that's what we went with. Here we've got a ribbon bipolar cell synapse. I don't know if you can see my cursor on the left side here. The ribbon synapse is a specialized kind of synapse seen in photoreceptors and bipolar cells. And it's making a connection onto a ganglion cell here to the left and an amacrine cell here on the right. And there's a gap junction in between the ganglion cell and the amacrine cell here. And so to confirm that this is in fact a gap junction and not an adherence junction, we do a tilt series in TEM and re-image it at 0.25 nanometers per pixel. And the technology that basically allows this is an older electron microscopy approach called transmission electron microscopy, but it works. This is a good time actually to start thinking about how one would go about making a graph of these relationships. So initially you think, how hard could this be, right? But then you look at the inherent complexity. This is the very first synapse in the visual system, right? So here's two cone terminals here, and they're making ribbon synapses onto an incredible tangle of processes right below the photoreceptors. How do you make sense of this, right? Robert's initial idea was that with this was that we'd capture some of the images, and then we'd draw a connection graph of these, and then we'd throw out the image data and move on to the next connection. Thankfully, for a lot of reasons, that idea did not last long, and we started realizing how complex the retinal networks were. And an emergent finding in the image data was how rich the image data was. So there's a lot more information in these ultrastructural images than just the simple connection graphs. And we'll get back to that in a minute. So in retina, if you imagine the simplest kind of network diagram, this would be it, right? Cone photoreceptors connect to bipolar cells, which then connect to amacrine and ganglion cells. And when amacrine cells feed back on bipolar cells and ganglion cells, and then themselves in these little nested feedback loops. So you need some way to describe the complexity of this network. And the problem is, it turns out that even describing these simple networks can get a little complex. In the 1970s, people were starting to look at network topologies. And Frank Harari came along and published this book called Graphical Enumeration, basically outlining how to determine the complexity of graphs. This equation is one example out of this book. It's a simple equation, but it can get a little complicated. So we can do a simple little thought experiment using Frank's equation. And if you can take three separate things, you can pretty quickly come up with 8 different ways that those 3 things can be put together. If you allow directed graphs, like a synapse between neurons, the number of potential combinations of these 3 things explodes to 64 different ways that you can plug these 3 things together. And just in case you don't believe me, here's graphical proof. So it turns out that if we allow reentrant pathways, like we have in neural systems, like the amacrine cells of retina or other inhibitory cells in brain, this explodes to 512 different topologies that can be represented by just 3 things, right? So what does this mean for neural networks? How many potential topologies are we talking about? To give you an idea of the size of the numbers, the number of seconds since the beginning of time is 4.0 times 10 to the 17th. Number of atoms in the universe is 10 to the 80th. If you play chess, the total potential number of moves that you can make in a chess game is 10 to the 123. And if you assume just 70 types of cells in retina, and we know that there's way more, that 10 to the almost 1500. And if you think about brain, that's with 1,000 cell types in brain, that's 10 to the 300,000th that you're looking at. And if you look at that Allen Atlas study that just came out a few days ago, they're estimating that there's at least 3,300 cell types in brain. So the number goes way bigger. What does this tell us? It tells us that these numbers are so big that you can't guess at what the network topology is, right? Let me give you another example. In 2017, there was this really fundamentally cool paper, and it's one of my favorites. Eric and Conrad asked a simple question in this paper, designing an experiment with a 6502 integrated circuit chip, which was a very common integrated circuit chip in the early days of computing. Could we probe the inputs and record the outputs just like we do for electrophysiology, and then use those data and reconstruct the circuit diagram. Right? So on the left is the Penn diagram for this integrated circuit, and it illustrates all the possible ways that one can interface with it. Right? It's a far simpler number of inputs and outputs than we have even with C. elegans that has 302 neurons, or 302 neurons. And on the right, we see the die size. The die size is approximately 4 millimeters by 4 millimeters. And that contains all 3,510 transistors addressable through these two address buses here and a data bus over here. And by probing and recording the outputs, it turns out we're absolutely unable to elucidate the wiring diagram without actually mapping the circuits. And it turns out we can't rely upon artificial intelligence to do the work for us in a piecemeal fashion either. These are 4 ganglion cells that I found from a paper from Heinz Vesely's work that I used as a training data set for a quick experiment on rules for how retinal neuronal architecture might form. And this was the result. The original 4 cells are shown here in the lower left, and the rest are clearly underparametized with some crazy stuff going on. But this experiment suggested to me that while we can't really understand the wiring rules by looking at individual components either, right, we need the network to inform us and act as additional discriminants or parameters that influence form and function. So all of this means that in order to understand the network, we absolutely have to map it, and we can't guess at what it means. Right? So this image is a map of 8,500 retinal neurons and their connection from one of our connectomes. How do you map this? Additional problems emerge, like, how do you interpret this mess? Clearly, it's more complex than we thought, and it's certainly more complex than the simple proposed retinal network. But how to pull this apart is another talk. So we've created a number of connectomes now, including this one, our first retinal connectome from a rabbit retina finished in 2009. We've also been annotating in a completed mouse retinal connectome, and we just finished assembly of a non-human primate retinal connectome, all 1/4 millimeter sized volumes of tissue at 2 nanometers per pixel resolution. Each one of these is a Herculean task that was only made possible through really an incredible team of people that I'm incredibly grateful for. But basically, our approach to volume EM allows for fast acquisition, high resolution, and embedding of metabolic data that helps us phenotype the cell types. So the color image at top is 3 probes for GABA in red, glycine in green, and glutamate in blue. This is critical to identify what the partners are. Because it's not just excitatory neurons versus inhibitory neurons that we're looking at, but what kind of inhibitory neuron or what kind of excitatory neuron. There's also a subtle yellow colored signal in this particular data set that's a physiologic marker that we'll get to later in the talk. So this is where I have to acknowledge some of the people on the team that makes this work possible. This is Jiahui Yang. She's our histologist who cuts all of our connectomes. Without her, we'd be lost. This is Matt Berardi, who runs our electron microscopes. He collects data on these things 12 to 16 hours a day and is responsible for the entire raw imaging throughput of the lab. And none of this would have been possible without this guy, James Anderson, Jamie. Jamie's our software architect. He created the tools and the database designs that we use to structure, contain, browse, and annotate all the image data in our connectomes. The Viking software package has made all of the inquiry, discovery, and circuit topology elucidation possible. And it's being used by a number of labs to look at their electron microscope data sets, including other connectomics efforts up at University of Washington in the Knights Library. So what's a connectome? Our connectome databases are more than just a bunch of registered images into a volume, right? We define a connectome as not just a collection of registered images or even a volume of image data, but a queryable database containing a relational graph of tissue in that volume, right? This is important because if the data are big enough and complex enough, If you don't have the means to interactively explore and query the data, then you're lost again, right? So we're tracking the connection graph as well as encoding Euclidean and geometric distances, synapse size and types, locations, organelles, cytoskeletal features, texture, and more. But this means that now you can find stuff. And for the first time in history, we can go back and forth between ultrastructural image data, morphology, connectivity, and even run statistical inquiries and queries on all of this completely interactively. It's an incredible hypothesis generation machine. So what has retinal connectomics taught us? We're still obviously making new connectome data sets, and we're mining our existing ones as fast as we can go. And we're extracting new data all the time. But there's been two major surprises, for me at least. One is that networks are more precise than we thought. And I'll give you two examples of incredibly high precision. And two, networks fail in more predictable ways than we thought in diseases like retinitis pigmentosa, age-related macular degeneration, glaucoma, that sort of thing. The first story of precision that I'm going to tell you about was largely due to Steve Massey. Right? As we were starting to go through some of the early annotations in RC1, our first retinal connectome, Jamie and I sat down with Steve at one of the Florida Arbos, and Steve suggested that a really cool network to chase would be the A2 amacrine cell network for lots of good reasons, especially if we could pay attention to gap junctions. And that advice turned out solid. So the funny thing is, when we first started exploration of these, we presumed that there'd be a certain amount of biological noise, right? That some retinal neurons would make wiring mistakes, but statistically errors would average out and everything would be good. Turns out this was incorrect. Retinal wiring is incredibly precise, even in complex nodal networks like the A2 amacrine cell network. And even though the A2 amacrine cell is one of the better studied cells in retina, our understanding of how complex this network was naive, right? The A2s, for those that don't know, are a really cool cell that bridges rod pathway input with the cone pathway, allowing mammalians to function from very dark environments to very bright environments. And this was the basic circuitry of the A2 amacrine cell pathway that was known up until around 2014, when, thanks to Steve's urging, we published this work, which summarized a ton of the work in the literature, as well as expanded our understanding of the precise circuitries around this pathway based upon connectomics approaches. So we knew previously that only cone bipolar cells drive ganglion cells, and that rod bipolar cells only drive amacrine cells. We now know that of the four classes of excitatory glutamate inputs, one here is from rod bipolar cells onto the arboreal network of the A2 amacrine cell down here. One comes from on-cone bipolar cells, again to the arboreal network. One comes from on-cone bipolar cells to the dopaminergic amacrine cells, which then make glutamatergic and dopaminergic contacts onto the neck of the A2 amacrine cells, and one from off-cone bipolar cells onto the lobules of the A2 amacrine cell here. We know that there's three coupling partners from short wavelength on-cone bipolar cells onto the arboreal network, and from on-cone bipolar cells onto the waist of the A2 amacrine cells, and then the well-documented arboreal network coupling. Dr. Crystal Sigolinsky in the lab has also, this is kind of fun, she's quantified the total potential current throughput via gap junctions of A2 amacrine cells. And it was stunning for us to realize that upwards of 25% of the total potential current through an A2 amacrine cell, at least in rabbit, is via gap junctions. So you can't ignore the gap junctions. If you want to understand what a network, retinal network looks like, you have to map them. And we also know from new work in the lab from Crystal that one of the classes of on-cone bipolar cells is from the CBB6 subclass. So again, these networks are precise. We've never seen deviations from these network topologies in any terminally differentiated normal tissue. that's clearly foreshadowing here. So speaking of Crystal Sigolinsky, much of the work in the lab on network topology is largely due to her efforts. She's been working in gap junctions as well as in a new area called mixed synapses that I don't have time to go into today, but she's working on that in collaboration with John O'Brien. But she's rapidly become a global leader in gap junctional biology through exploration of connectomes. And I might argue that because of the way that connectomes are structured, she might have looked at more gap junctions than anybody else on planet Earth at this point. It's kind of fun. So one of the areas of network precision that Crystal's been chasing has been the role of gap junctions as cell classifiers, right? Up top here, Crystal's arranged the on-cone bipolar cell classes here in morphology and lamination, just like the work from Dick Maslin, Heinz Vesely, and Silke Haverkamp, who based their work on morphology and other analyses. But Crystal's work has basically shown the incredible precision of gap junctional connectivity that defines additional cell classes. And here's one specific example within the CBB5 on-cone bipolar cell class, where they exhibit within class homocellular coupling to create these extensively coupled sheets where they make gap junctions at the tips here of all of their dendritic arbors. And without fail, the interesting thing is all CBB5 cells make gap junctions. So if you look at every opportunity in these cells, they all make gap junctions. And in each one of these B, C, D, E, F are sort of zoom ins showing the ultrastructure, and the prime number here is a 0.25 nanometer per pixel a zoom in confirming the gap junction. So you can debate whether or not this is a syncytium, but that's going to depend on modeling efforts that we need to get to. But you might sort of imagine that if this were to happen in the off layer, it might be a reasonable retinal mechanism for the looming response that so many folks have been talking about lately. In contrast, other cone bipolar cell subclasses decline in cell homocellular coupling, right, despite opportunity, right? So the CBB6 on cone bipolar cells always refuse contacts. So they'll make tip-to-tip communications, but these tip-to-tip communications as seen in C always end up in adherence junctions, whereby the same CBB6 cells are more than perfectly happy to make gap junctions with the A2 amacrine cells next to them, right? And again, the important thing here is the resolution. If we did not have the resolution and the ability to re-image these, we could not have told you that these were in fact adherence junctions and these were gap junctions, and we wouldn't have been able to discover this. If you want to know more about this, Crystal's got a great paper in JNeurosci a couple of years ago. So while we're chasing the normal networks found in retina, we're also chasing the idea that these networks are altered in neurodegenerative disease. And the early assumption that I had was that when neural networks fail, they'd fail in chaotic ways. And as I alluded to earlier, this assumption was also incorrect. I don't have time today to go through all the details of retinal remodeling, but just know that it's been a 2-decade-long process of documentation, finding that the universal response to photoreceptor cell loss in adult terminally differentiated retina is a dynamic negative plasticity that alters neurons, glia, circuitry, structure, function of both the sensory and computational retina. And this corruption basically alters the retina's ability to detect and compute information required for vision. And when you look at the end stages of the disease, it certainly does look chaotic. The end stage of the disease is basically, it was my first exposure to retinal degeneration. Ann Milam sent us these aged human tissue samples from a patient with retinitis pigmentosa, and this is what we saw. So this is a triple label of taurine, glycine, and glutathione in red, green, and blue color channels. And it looks nothing like normal retina. And at the time, I didn't understand it, and I assumed that it was some sort of like weird neoplasm or cancer. And we set the slides aside. And then something magical happened. Jeannie Frederick came by the lab and casually mentioned that she and Wolfgang had some old GHL mice, mutants. It was a model for retinal degeneration that they forgot about in the back of the cage. And they got quite old, and in this case, like 2 years old. But she wondered, would I be interested in looking at the retinas? Otherwise, they were just going to sack them, and because they were too old for their experiments, because they didn't have photoreceptors anymore. And this is important because back then, assessments of retinal status were based upon standard histologic sections. And if we quantified the data, we'd make spider plots of the thickness of the outer nuclear layer like this. These data are from Luna Nash's lab that document how the outer retina thins over time. And even now, traditional histologic preps like tall blue are the common currency, right? In much of the literature that gives the ability to discriminate layers and count the numbers or layers of photoreceptor cell bodies to do quantitative measures. But what happens is that when the photoreceptors are gone, the experiment's over for many folks, especially for those who just study photoreceptors, which is why Jeannie and Wolfgang were going to give those GHL mice to me in the first place. But lots of assumptions have been made about this, which at least here in this particular TGA9N mouse retina from Jason Chen, the neural retina at least looks okay, leading many folks to make assumptions on the level of intactness. So when it came time to intervene and cure blindness, strategies were developed based upon this level of knowledge to rescue vision. But these are the retinas from the GHL mice, right? Again, labeled with taurine, glycine, and glutathione in red, green, and blue. And it looks shockingly like the human RP retina. And again, it looks chaotic. And so, my assumption was that this was chaotic. And this was 1999, and I got really excited, and I ran to the library and started digging around and found this paper from, turns out, Ann Milam and Bob Ferris. And this paper basically started to influence my thinking on retina becoming a dynamic negative plastic environment in response to photoreceptor deafferentation. So much of the early work and the very first findings of retinal degeneration was from Enrique Stratoy, I mentioned earlier, but also Yinni's Bob Ferris, who, after he did his PhD working with Steve Fisher and Jeff Lewis in retinal detachment, went up the coast to Ann Milam's lab to do a postdoc looking at photoreceptors and neurons sprouting. And that's when we were sent the human tissue that started this whole thing off. I spent a lot of time looking at late stage retinal degeneration, but we started looking at the earlier stage of retinal degeneration, and including changes that predate photoreceptor cell loss, right? So photoreceptors get stressed and they start to degenerate, but we were asking what happens in the neural retina underneath in response to that. And so these data are data from wild type in our D1 mouse retina looking at protein kinase C alpha. And on the left, you see wild type, and on the right, you see the RD1, I think 60 to 90 days old, 60 to 90 days of age. And you can see up here, there's a dramatic loss of these beautifully sort of tufted dendrites in the RD1 mouse. We also started using gene gun ballistic, like dye based labeling to image single rod and cone bipolar cells in wild type and RD1 mice, and looking at trying to quantify the earliest changes in dendritic morphology. And we did a lot of that, but we were also wondering about the physiology, right? When do photoreceptors and bipolar cells stop processing information in retinal degeneration? And to get at the physiology, we can probe glutamate channel function in tissues by combining excitation mapping and spatial metabolomics imaging, right? Where we can visualize neurons excited by ligands or light pathways. And in this case, we used a papillonovus retina, non-human primate ex vivo stimulated by 25 micromolar AMPA in the presence of a glutamate channel permeant marker called AGB. So here we've got simultaneous imaging of the GABA in the red color channel and glutamate in the blue color channel. And if we add in AGB in the green color channel, it shows that characteristic sets of neurons, horizontal cells, off bipolar cells, amacrine cells, and ganglion cells can be activated by this pharmacologic drive. Again, in this case, in the presence of AMPA. And if we zoom in, we can see that AMPA strongly activates horizontal cells and off-cone bipolar cells while not activating Mueller cells, rod bipolar cells, or on-cone bipolar cells. And this is just the AGB channel to show you that this is quantifiable. And you can, there's other pharmacological means. You can drive retinas like this with canic acid or NMDA, with the idea that you want to pull apart the retinal populations and see which one responds to certain circumstances. So it was at this point that Minio Kondo and Hiroka Terasaki developed their transgenic rabbit model of autosomal dominant retinitis pigmentosa. And Minio came up at ARVO and offered it to us as a model of large eye RP, and would we be interested in collaborating with them? This was really exciting because we could start using all the tissue in these large eye models and do the same thing that we'd been doing in non-human primate and mouse models of RD, as well as later on in human RD. And it turns out there were a couple of things that we didn't understand after looking at the physiology of these retinas. One of which was an apparent reprogramming of bipolar cells in mouse, human, and rabbit retinal degenerations that again occurs prior to photoreceptor cell loss, right? And before any bipolar cells have been lost. Effectively, the whole pharmacological profile of bipolar cells is altered. And here we've got 3 panels of rabbit retina with horizontal sections. So you're looking all FOS through the bipolar cell and amacrine cell layer. And we've got IGLUR-driven off bipolar cells in green, IGLUR insensitive on-cone bipolar cells in red, and rod bipolar cells in blue. The wild type is on the left panel, and the 3-month-old transgenic rabbit retina is in the middle, and the 10-month-old transgenic rabbit retina is on the right. And what we found was that bipolar cell types started switching their phenotypes to become more off-like, as if rod bipolar cells and perhaps some on-cone bipolar cells were starting to express IGLUR receptors. We had no idea what that meant or where that reprogramming was occurring, particularly if the dendrites from the rod bipolar cells were starting to truncate, like we previously showed. So another thing that we didn't understand was that changes in glycine content in bipolar cells were happening. So this is a horizontal section through the bipolar cell and amacrine cell layers of our first retinal connectome with co-localized glycine label on them, overlain on top of the ultrastructure, right? So this is a quantitative signal showing variation in glycine content of amacrine and importantly, the bipolar cells. This is another example of precision, by the way, in that normally in wild type tissues, glycine concentrations are robust, stable, and unique in every cell type. And normally your rod bipolar cells come in 3 types, rod, on-cone, and off, in roughly equal proportions, right? And in normal tissues, of the bipolar cells, only the on-cone bipolar cells have any glycine in them, right? We don't know what on-cone bipolar cells are doing with that glycine, but we do know that it comes from gap junctional coupling with A2 amacrine cells that we just talked about a few minutes ago. So this is true in rabbit, mouse, non-human primate, human retina, as I'm sure in other mammalians as well. This is an image of normal mouse retina with GABA in red, glycine in the green color channel, and glutamate assigned to the blue color channel. We can just look at the glycine channel out of this data set. And if you pay attention to the glycine in the bipolar cell layer of the retina here, you see variation that's reflective of a normal expected distribution. Among bipolar cells, only the on-cone bipolar cells have any appreciable glycine in them. This changes in transgenic mice that are starting to experience photoreceptor cell stress, but prior to photoreceptor cell loss, right? In this mouse, we're probed for GABA, glycine, and glutamate, again assigned to red, green, and blue. But there's an alpha channel here that represents the glutamate channel permeant marker, AGB, that we just talked about a minute ago. And this shows something really interesting. There's way more glycine in the bipolar cell layer than one would expect. So this is a mathematical AND of the glycine and AGB channels here. But it shows us that in this bipolar cell layer, there's way more glycine present. And we had no idea why this was. But you know, we were debating all sorts of things. You know, was metabolism being changed? Was, you know, were we unmasking some sort of metabolic profile? We didn't know. But it turns out Pathoconnectomics, or the application of connectomics technologies to pathological tissues, came to the rescue. And this is where Becca Pfeiffer shows up. Becca is now a research assistant professor in my lab. She's chasing Mueller-Glia as part of her independent career. But when she was a postdoc, she took upon the daunting task, if you will, of heading up our Pathoconnectomics initiative. And in this case, we used retinas that are undergoing retinal degeneration from that transgenic rabbit model of autosomal dominant RP that we got from Minio and Hiroko. Basically, we want to know precisely how these retinal networks are changing. And as it turns out, it's more complex than just the photoreceptors dying. But as I've hinted at, these changes are predictable and they're stereotyped, right? And again, this very first pathoconnectome that we made was made at the earliest time point when we still have photoreceptors, and when the bipolar cells are starting to switch their phenotypes, and when they start developing all the glycine in them. So this early stage of retinal degeneration is important. The observed changes to glutamate channel expression and Mueller cell reactivity begin prior to photoreceptor cell loss. So the first obvious target for us, at least, was to look at rod bipolar cells in these massive data sets. So starting up high, we wanted to look at bipolar cells to figure out if there was any ultrastructural evidence for why they might be increasingly expressing IGLUR receptors. And as Ann Milam's team and Enrique Stratoy showed all the way back in 2000, rod bipolar cells and horizontal cells are some of the first cells to start sprouting and going awry. So this documents normal rod photoreceptor to rod bipolar cell contacts, even if they're a little truncated. But it turns out these normal connections at this early stage in retinal degeneration are very much in the minority. And here's where stuff gets interesting. The majority of bipolar cells are doing abnormal stuff. And sure enough, the first cell that we looked at, a rod bipolar cell down here in blue in this image, had plugged itself up into a cone photoreceptor pedicle, and was at the receiving end of a cone photoreceptor ribbon, right? So what's this mean for function, right? What glutamate receptors are present there? From the physiological data that we just talked about earlier, a safe bet is that this rod bipolar cell has switched its glutamate receptor expression, is now expressing IGLURs rather than the normal IMGLURs that were shown by Nakajima back in the early 90s. Or was that Malcolm Slaughter? I can't remember now. Somebody correct me in the Q&A. But regardless, rod bipolar cells are now on the receiving end of cone photoreceptor signals and might very well be expressing IGLURs in their dendrites rather than the IMGLURs that they normally express. So the next 3 or 4 slides will explain the glycine content that we found in cells, and then we'll wrap up, right? So looking at the first synapse from photoreceptors to bipolar cells, we then wondered what the rest of the rod bipolar cells were doing. And we were wondering what their synaptic inputs to A2 amacrine cells would look like. And if you remember our A2 conversation earlier, we know that rod bipolar cells normally make conventional synapses on the terminal arbors of A2 amacrine cells. but we found something interesting. Again, normally the cell classes that make gap junctions with A2 amacrine cells are on-cone bipolar cells. These gap junctions from on-cone bipolar cells to A2 amacrine cells in the arboreal network and waste are literally physical coupling, right? And they make a pore that small molecules like glycine leak through. So you can literally segment out those bipolar cell populations just by looking at the glycine signal in normal tissues. So you can imagine how surprised we were by absolutely confirming rod bipolar cells making gap junctions with A2 amacrine cells down in the arboreal branches where they'd normally make just synapses, right? And if you zoom in, you can confirm the pentolaminar structure of gap junctions here in a rod bipolar cell receiving synaptic input from rod photoreceptors. It is making synapses down on the terminal arbors of the A2 amacrine cell, but now it's also making gap junctions with A2 amacrine cells in the same place. And then we found off bipolar cells also making gap junctions with A2 amacrine cells up on the neck of the amacrines of the A2s, where they normally make IGLUR mediated synapses. And so this network topology certainly explains where the glycine is coming from in all bipolar cell classes. It's leaking through the gap junctions, right? Just like it does for on-cone bipolar cells. But this also raises all sorts of questions, right? Since gap junctions, it's sort of, it takes two to tango with gap junctions, right? Where are the connections coming from in the rod and the off bipolar cells, right? Have they always been able to make gap junctions that they just don't normally? What's the mechanism behind this unmasking, right? What does this mean for where current can flow in the system? Have we made a de facto syncytium where current sort of can spread throughout the system? Botir Tsagdalev showed a few years ago that these retinas oscillate wildly, and if you block gap junctions, the oscillation is damped. Maybe this is the ultrastructural mechanism for that. At any rate, study section just met a couple of days ago on my renewal. Hopefully they score us well enough to continue studying these questions. Let me wrap this up here by saying, for those interested, we have a number of retinal connectomes now. We have a rabbit retinal connectome. We've got a full thickness mouse retinal connectome, a full thickness non-human primate retinal connectome that we're just wrapping a database up for, and these three pathoconnectomes. All of them are at 2 nanometers per pixel resolution, and the raw data and our annotations are all open source. We're developing code that allows folks to explore and annotate on their own, as well as create some new AI-assisted tools that will help with annotation. But because all of these volumes and existing annotations are open source, they're available now for you to explore and plan your own experiments around. If you're interested, get a hold of me for specifics for like URLs and the software that you'll need to download from our websites to explore them with. And then I should add that there's more in these data sets than just neurons. Dr. Pfeiffer, who I previously mentioned, is chasing Mueller glia as part of her independent career. And she's tracking the interactions with synapses involved with Mueller glia as they undergo retinal degeneration over time. Right? Because we have volumes from multiple species, we now have the opportunity for comparative connectomics, and Dr. Sigolinsky has been heading up some of those efforts. And we now have a student in the lab who's chasing the vasculature of the mouse retina, where we've got glia, parasites, endothelium, and all the neurogliovascular data for labs who are interested in exploring. And finally, let me just acknowledge some of the undergrads in the lab who made this work possible. All 4 of these women are first-generation college students. All are from underrepresented populations in more ways than one. Jessica and Jabika are now medical students. Isabel is now a nursing student, and Selena is working on the vascular project in the lab. She's currently applying to medical school soon. And Megan Kroom is a grad student in the lab who's taking an aging approach to these tissues and is chasing down proteinopathies with retina as a model for CNS neurodegeneration. The following funding here has made this possible. We're grateful for funding from the NIH and the NEI, the NSF, an unrestricted grant from Research to Prevent Blindness, and an incredibly generous grant from Gabe Newell, which could not possibly have come at a more opportune time. And for that, I'll always be grateful. I'll just wrap up by saying I'm leaving for Torremolinos for the RD meeting on Sunday. I hope that if you're there, we'll have a chance to catch up on any questions that don't get answered today. Thank you.
Thank you, Bryan, for this intricate talk. And we will start with a couple questions from the room here with Zach.
Hi, thank you so much for that really interesting talk. So I had a question. Is there some interest in assembling foveal connectomes next in non-human primates?
Yeah, so like there's a lot of foveal stuff happening up at University of Washington, right? So Dennis Dacey and the Knights are putting together foveal connectomes. For me, parafoveal retina is more interesting scientifically, but certainly, yeah, that work is being done up at University of Washington.
Are you also interested in kind of integrating signaling data into your connectomes next, kind of assembling, signaling pathways de novo and correlating that with, in vitro or in vivo data?
Absolutely. So for us, this connectomics, it's just a framework, right? And we've got to hang all of the ePhys data, all of the MEA recording data, all of this stuff sort of on this framework so that we can start actually modeling how information flows through these networks.
Do you think because you have these like very, very complex volumetric reconstructions that kind of the architecture will successfully recapitulate those signaling networks just because you have all the connections mapped in theory? That's
the hope. And we're working with like Gianluca Lazzi down at USC to develop some of this. So the problem with a lot of the modeling out there is that like a lot of the software tools for neuronal modeling, they presume much simpler systems, right? So we're literally sort of in ball and stick sort of worlds with single compartment models on a lot of these. And we're starting to push multiple compartment models where we model the soma as one compartment and the initial segments of dendrites and axons as additional compartments. But almost none of the modeling software out there takes into account morphology or some of the more Euclidean and geometric distances involved in that. So it turns out it's tricky, but it certainly needs to be done, and we hope that these data can sort of inform those modeling efforts.
Thank you.
And Emma?
Hi, Dr. Johns. Beautiful talk. My name is Emma, and I'm interested in diabetic retinopathy. That's my field of research. So I'm very happy that you are working on the vascular connectomics now. Yeah, that was one of my questions. My second question is, you show us on all your work, basically topical, regional connectomics. Do you envision any distant connectomics, between the retina and other cells in the body, distant cells in the body, and vice versa?
So a lot of this is determined by the laws of physics in that sense, in that, if we're interested in synaptic level stuff, your data sets can only get so big. There are ways of bridging this in doing mesoscale level connectomics where we can look at projections, and you can do that at the light microscopy level and also ultrastructural level, and there are ways of doing coarser resolution level projections That work absolutely has to be done. It's being done in some cases. There are certainly the Drosophila retinal or the Drosophila connectome has just been done, and there's a new microphragma connectome that's being worked on out of Europe, and so those are taking smaller organisms so that they can take an ultrastructural approach and get entire sort of circuits beyond sort of the visual system. Yeah, it's important. It needs to be done. It's technically very difficult to do it at the resolution that we're operating at.
All right, thank you, Emma. And we go to Daniel. And then Steve.
It's nice to see your face, Daniel.
Yeah, good to see you, Brian. Excellent talk, beautiful images as usual. Thank you very much. Can you talk a little more about the differences between rewiring or remodeling of the retina in RP versus AMD, in geographic atrophy in particular? Do you have any input?
Yeah, so we did a paper in 2016 where we were looking at dry AMD, and basically the rules are the same. Whenever you deafferent a complex neural network that's terminally differentiated, the downstream elements have a couple of decisions to make. Cells still need to maintain calcium signaling. They still need to do gene regulation and protein expression work. And they require afferent inputs in order to define their activity. If they don't get them, then we believe that the common pathways for responses are the same, right? So we saw the same kinds of pathologies in AMD tissues, in human AMD tissues, that we saw in human RP tissues and models of RP.
But they look very different in OCT. In AMD, in geographic atrophy, you see very nice preservation of layered structure in the retina, and it's local, degeneration, obviously, versus global. In RP, it's indeed very messy, unlayered structure of the retina at a late stage.
So they're both regional, right? So RP, you get these regional patches where you've got bad retina right next to sort of some good retina. And over time that spreads and it merges. And the same is true for AMD, right? So we see regions, particularly under like drusen, where, in fact, the first defect that we see is in retinal pigment epithelia. So if you do a metabolic mapping of RPE cells, those metabolic signatures should be absolutely identical in every single RPE cell around, because RPE cells are coupled via gap junctions, and those small molecular metabolic signals equilibrate through. And the very first defect that we see in geographic atrophy or dry AMD is a metabolic bricking of RPE cells. And when that starts happening, we see indications of photoreceptor cell stress, opsin delocalization in cones, and then the downstream elements. We haven't done the pharmacologic studies in those yet, and we haven't done the physiology in those cells, but we see the same sprouting, we see the same Mueller cell reactivity, we see the same alterations in connectivity in AMD that we see in RP. I mean, I can't tell you about time progression yet, because we haven't really done aggressive series of, there are no good models of human AMD with drusen that we've been able to, find in our systems. And so we've been dependent upon human tissues to do that.
And then Steve, are you there?
I am there.
We'll have final comments from Seth Blackshaw.
In fact, I'm here. So Brian, fabulous story. I've been following the evolution of this story for well over 2 decades. I learned a lot of new things today. One of the things I learned is this incredible plasticity of the neurotransmitter receptor subtypes that can occur when you have these kinds of remodeling. 2 questions. One, you showed us an example where a rod photoreceptor cell is making inappropriate contacts with amacrine cells which requires that rod cell to really extend its body into a layer of the retina that it should not be in. But I know that you've published previously also on a different kind of plasticity, and that is ectopic reorganization of cells where they literally leave their normal neighborhoods and go elsewhere. What happens to the physiology in those cases?
Yeah, so we have less, we have fewer ePhys experiments in the later stages of retina because of the large amounts of cell death and the complexities associated with identifying specific cell types that late in the game. So in late stage retinal degeneration, when cells are moving around, sometimes it can be hard to figure out if they are in fact looking at spatial metabolic mapping. Is that a bipolar cell anymore? Is that an amacrine cell anymore? Is that a ganglion cell anymore? There's extensive coupling that starts happening, and GABA and glycine start leaking through into other cell classes. And so it can be hard to figure them out. And so that's a question that at least in late stage retinal degeneration, we don't know. For us, we were more interested in finding the earliest changes. And so that's where most of our work went.
Good, thanks. And then lastly, you ended up talking about some emerging work on the Mueller glia and their interactions. And as probably everybody on the call knows, that when the retina starts to degenerate, it's actually even before you can see any overt histological changes, the Mueller cells begin to hypertrophy, their GFAP, their intermediary filaments expression skyrockets. And then as cells start to degenerate and space starts to open up when cells are dying, the glial cells expand, the hypertrophy, right? And so there are opportunities to have inappropriate metabolic interactions between the glia and the remaining neuronal populations.
Right. So I didn't show the ultrastructural data in our second pathoconnectome and the third pathoconnectome yet, but normally, Mueller cells are listening at every single synapse, right? And there's a complex biochemical dependency of neurons upon glia to cycle glutamate and glutamine and provide some of the carbon skeletons required for normal metabolism and neurotransmission. And so one of the questions is, when Mueller cells start hypertrophying and changing, do those relationships with synapses change over time? And that's new work that we're just starting to chase now. That's going to be Becca's career focus on that.
Good. Thanks so much. Vaya con Dios. See you in Torremolinos.
Yes, sir. Thank you.
Right. And now we are looking forward to final remark by Seth Blackshaw.
Okay, well, Bryan, I think you've given an amazing and very convincing explanation for why this kind of large-scale connectomic analysis is absolutely essential for full understanding of neural circuitry, and why the retina is the ideal system for doing these kind of connectomic analyses because of its cellular simplicity, lack of experience-dependent plasticity, and disease relevance. And you really have set the current standard in the field. So it's a real pleasure to hear. And I think some of the key issues that at least occur to me going ahead is, first, can we use this kind of this high resolution morphological and connectomic data? Can we integrate that with the molecular data that we have, and to identify connectomes for newly identified functional subtypes of bipolars, amacrines, and ganglion cells? Also, the functional significance of these new connections that you've identified in both the healthy retina and in the pathoconnectome. It's likely that many of these plastic changes are adaptive some may be truly pathological, but some may be helpful.
Absolutely.
And this is very important as far as potential therapy goes. And then lastly, moving from the pathoconnectome to really the therapeutic connectome. How do interventions that improve outcomes change the connectome? And in particular, from what very dear to my heart, what happens in regenerative cases? So if cells are replaced by transplantation, or if nuclei are generated from, or if new neurons are generated from euglia, are they able to integrate? How do they do this? And how does that differ from endogenous cells? So again,
yeah. Thank you. No, I mean, there's a tremendous amount of promise here for this, and to sort of address these things. Yes, the pathoconnectomes are important, and they sort of reveal the changes that absolutely do occur. They're sort of pointing the way towards biological rules or rule sets that are invoked. These rule sets appear to be directional. They go in one direction, but as we know from like Daniel Kirstensteiner's work and everything, they might be modulatory, right? We might be able to learn how to manipulate what biology is telling us and use some of the features that the negative plasticity is telling us to figure out how to modify that. Can we, I mean, again, this whole question on gap junctions, do rod bipolar, I mean, can rod bipolar cells normally make gap junctions, right? I mean, what's happening when that happens, right? What does that mean for normal biology? Can that be tuned? Can we stop them from making gap junctions, right? Are there lessons that retina can teach us that are generally applicable to CNS disease like Alzheimer's? Those are, I mean, the cool thing here with retina now is that we've got a potential early model for Alzheimer's, right? There are no good early models for Alzheimer's, but retina may sort of point the way, because we know that in late stage retinal degeneration, we start seeing the same proteinopathies that people are chasing in Alzheimer's. So there may be an entire sort of biological rule set here that we can learn from. And the cool thing now is that because these changes are so stereotyped, we can do the same experiment again and again and again. We don't necessarily have to generate huge retinal connectomes, but we can identify specific cell classes, and we know what they're going to be doing through these pathoconnectomes, that we can generate smaller ultrastructural data sets that don't have to be these monolithic connectomes, and we can start bridging them with expression data and with seq data and with all of the other tools to start sort of teasing these biological rules apart. And then from like a developmental perspective, man, I mean, we really need some developmental connectomes. Organisms like Whaley's ground squirrel or Steve Drewry's ground squirrel, right? That teach us that there is a plasticity in hibernation, that the photoreceptors can degenerate, the synapses can unplug and float free, and it looks just like it does in human RP. And then they come out of hibernation and everything reassembles and it functions just fine, right? Or in zebrafish, when we lesion zebrafish retina or damage zebrafish retina, there is that population of stem cells that hides out in the Mueller cell population. It dedifferentiates and appears to recapitulate a normal functional retina, right? That's a more complex retina than ours. How is it doing that? And some of those connectomes would be really fascinating. Yeah, no, it's an exciting area. I kind of think of sort of volume-EM or connectomics, kind of where genetics was 40 years ago. It's early stages. We need a lot of sort of shared tools, but there's a lot of experiments that people can do around these frameworks.
Okay, let's thank our speaker.
Thank you so much. It was nice to see your face, Seth.
Yes, and you.
All right.